Welcome to the first in this series of lectures on amino acid metabolism. In this lecture, I'm going to define the basic structure of an amino acid. I'm also going to illustrate the difference between essential and non-essential amino acids. I'll describe the pathways for protein degradation, the two main ones in your body, and I'll also introduce the concept of nitrogen balance. But to start with, let's begin by looking at the structure of an amino acid. So here is the basic form of the amino acids in your body. You can see that there's a carbon backbone, an amine group with nitrogen in it, and crucially, this R side chain. It is this bit here that defines the difference between the variety of amino acids in your body. Now, this variety of amino acids actually only extends to 20. Now, these are mixed together in a multitude of combinations to form the proteins in your body. But fundamentally, these proteins consist of mixtures of these 20 different amino acids. Now, in the previous slide, I was describing how this R group here defines the difference between each of these 20 amino acids. And here's a nice example of what I was talking about. Here you can see alanine, and here is its R side chain. Here's L-cysteine and L-lysine. So in each case, the basic backbone of the amino acid still exists, and it is simply these R side chains that differentiate them. Now, to form a protein, these amino acids are joined together via peptide bonds. Two of these form a dipeptide, 3 amino acids form a tripeptide, and so on. So these chains of amino acids represent the primary structure of a protein. These chains are linked and folded into quite complex structures to form the functional protein. And we call this the tertiary structure of a protein. But the sequence of amino acids that are used here to build these peptide chains are quite carefully controlled, as they do have a big impact on the function of the resulting protein. And of course, the map telling you which amino acid to grab next and include into the makeup of that protein is sitting embedded inside your DNA. Ultimately, your DNA simply codes for one protein at a time, telling your cells how to construct them. So therefore, one of the key roles of amino acids in your body are to act as building blocks for proteins, and therefore they have a major function in protein synthesis. However, during starvation, the carbon backbone of these amino acids can also be used as a source of energy, or as a source of glucose via gluconeogenesis. And in addition to that, amino acids act as precursors of specialised compounds, including things like porphyrins, hormones, and other signalling agents within your body. Now, when you listen to nutritionists, they'll refer to these amino acids as either essential or non-essential amino acids. This definition is really quite simple, and it simply links to whether or not you can endogenously synthesize that amino acid or not. If you can make it inside your own body, then it's considered to be non-essential. However, if you can't endogenously synthesize it, then it's an essential amino acid. And for essential amino acids, there is only one way to get hold of them. You've got to eat them. It's for this reason that ration formulations in animal nutrition has to be carefully controlled to ensure that all of the essential amino acids are included in the diet. This is particularly important for monogastrics like pigs. And just so that you've got access to it, here's the list. These are the essential amino acids on the left, and these are the non-essential ones on the right. So in effect, you can synthesize these endogenously, whereas you have to get these ones through your diet. And you'll note, if you add up the two columns, you get a total of 20. For the non-essential amino acids that you can synthesize endogenously within your body, here's a list of their synthetic reactions to produce them. But don't panic, I've included this list more for your information. You don't actually need to learn this for the exam. I would like to highlight the top 3 ones though. The first one is alanine, and you can see that if you transaminate or add a nitrogen group to pyruvate, you end up with this amino acid. The next one down is aspartate, and again, if you transaminate and add an amine group to oxaloacetate, then you end up with aspartate. And the last one is glutamate. So in this case, if you add an amine group to alpha-ketoglutarate, you end up with glutamate. Now it's essential to maintain a pool of free amino acids, and animals have 2 key sources of these. The first is a fairly obvious one. It's through the diet, through digestion and absorption. And the second is through endogenous protein degradation. So in effect, you can break down your own proteins to replenish your amino acid pools. Now it's quite crucial to point out here that animals don't necessarily store protein as a pool or a depot of energy in the way that they store, say, glucose as glycogen. So generally, proteins are not a key energy depot. However, when dietary protein supply is limited, it's in this instance that endogenous proteins are catabolized to replenish those amino acid pools. Animals have 2 key pathways for protein degradation. The first is the ubiquitin proteasome pathway. This is an ATP dependent and so energy consuming process, and it's non-lysosomal, so it means that it doesn't occur in your lysosomes. This is the major route of degradation of muscle protein in mammals that releases amino acids, particularly during starvation. It's also involved in the degradation of critical regulatory proteins, and it's also crucial for degrading highly abnormal proteins that may arise from mutations or post-synthetic damage. The second pathway for protein degradation is the lysosomal pathway. This is the major pathway for protein degradation in the liver, and it involves a range of cathepsin enzymes within the lysosomes. To give you a bit more detail about the ubiquitin proteasome pathway, this essentially involves the ubiquitinylation of target proteins. So you can see in the diagram here, the target protein has a molecule of ubiquitin attached to it. This costs an ATP, and this ubiquitinylation occurs, creating a chain of ubiquitin molecules. So we call this a ubiquinated protein. And this has been marked for degradation. These proteins, these ubiquitinated proteins, so those that have been marked for degradation, are then endocytosed within the proteasomes whereby they are broken down into their constituent peptides. So we can contrast these 2 pathways. As I said, the ubiquitin pathway is targeted towards abnormal, short-lived or regulatory proteins, and this involves the ubiquitinylation of these proteins, which are then degraded in the proteasomes, and it costs ATP, so it's an ATP-dependent process. Now generally, this pathway is non-regulated, so it's got this underlying relatively constant rate. The lysosomal pathway is targeted towards extracellular and membrane-associated or long-lived proteins. This, of course, occurs in the lysosomes. It's mostly an ATP-independent process, and in this case, the proteins are internalised within the lysosomes and degraded by cathepsin enzymes. Now, this is a strongly regulated pathway. So let's have a look at the factors that influence or regulate the lysosomal pathway. Firstly, there are a number of hormonal regulators. And particularly for these 2, there's good logic behind their impact. When you're starving, glucagon is released, And this has a positive effect on the lysosomal degradation of proteins. So in effect, this is the way in which starvation can speed up protein breakdown, making amino acids and their carbon backbones free for gluconeogenesis. In contrast, insulin has a strong negative effect on protein breakdown. And again, this makes good sense. When you're well fed, you actually want to synthesize protein. You don't want to be breaking it down. And glucocorticoids, the long-term stress hormones, have a positive effect on this pathway. So they increase the breakdown of proteins, but in a more subtle way. There are also some nutritional regulators. So the two amino acids, asparagine and glutamine, when these are in high concentrations, they'll have a strong negative effect on the lysosomal pathway. And in some ways, this is a good indicator of the fact that you are well fed, particularly with a good source of protein, making these two amino acids high in concentration, slowing down protein degradation. So you can start to see how nutrition, and more specifically the intake of proteins, can influence protein turnover. When dietary protein intakes are high, you'll have a corresponding increase in plasma amino acid concentrations. The enzymes involved in catabolizing these amino acids have a fairly high Km, and therefore they swing into action only when these plasma amino acid concentrations are high. Of course, having a high Km also means they'll have a high Vmax, and therefore you get a corresponding high rate of amino acid catabolism within the body. Alternatively, when protein intakes are low, you'll have a corresponding low concentration of plasma amino acids. Now, the enzymes involved in protein synthesis, in particular the amino acyl tRNA enzymes, have relatively low Km values. Therefore, they can function when amino acid concentrations are quite low. They still get access to the substrate that they need to build protein, albeit at a slower rate. But this underpins a relatively constant activity and a relatively constant rate of protein synthesis. Nonetheless, don't lose sight of the fact that if you do have low dietary protein intake, you will see a general depression in the rate of protein synthesis within the body. So this leads us to the concept of an amino acid pool, which your body is trying to maintain at a constant level. Consuming dietary proteins adds to this pool. Protein synthesis within your body detracts or takes away from this pool, but protein degradation adds back into it. The synthesis of non-protein molecules takes away from this pool, as does the deamination of amino acids, so that you can release their carbon backbones, enabling you to either make glucose, generate energy, or synthesize fat. I'll talk about this in the next lecture, but it's crucial to remember that when you deaminate amino acids, you also release nitrogen, which has to be synthesized into urea in the liver and then excreted through the kidney so that the nitrogen doesn't poison you. So we refer to this interplay between amino acids and protein as your body's nitrogen balance. Here's an example for a 70 kilogram adult human. In this case, their intake of amino acids from their diet is equivalent to about 100 grams. So this is the I value. And this adds into the amino acid pool, which consists of about 500 grams of free amino acids. Protein turnover is constantly taking place. So there are 400 grams of protein being synthesized, which is the S value, and another 400 grams being broken down or degraded. So that's the D value. So in this case, given that synthesis and degradation of proteins are in balance, we need to maintain the balance of our amino acid pool, and therefore 100 grams is broken down or excreted after deamination. So in this case, to determine your body's nitrogen balance, you simply add I plus D, and to be in nitrogen balance, it has to equal S plus E. Alright, so let's look at some scenarios. And the first one is starvation. So of course, in this case, there is no dietary protein intake, adding to the amino acid pool. Now, when you're starving, you'll be deaminating amino acids so that you can release their carbon backbone to make glucose, produce energy. Hence, these have all got big fat red arrows. So to replenish your amino acid pool, you have to increase your rate of protein degradation from your body protein. So it gets another big fat red arrow. And therefore you manage to maintain the size of your amino acid pool. But in effect, in this scenario, you would be in negative nitrogen balance. Now consider a low protein diet. In this case, there's plenty of energy kicking around in the body, but specifically dietary protein intake has been reduced. Because you're well fed with plenty of energy kicking around, your protein turnover, so your body's synthesis of protein, will be in balance. So you'll be synthesizing body protein at the same rate as you're degrading it. And therefore, so as not to diminish your amino acid pool in your body, you slow down the rate of deamination of these free amino acids. Now, this slowing down is regulated in a very simple fashion. It occurs simply through the Km of the transaminases and the deamination pathways involved in this step here. So these are all high Km enzymes that are in action, and therefore when the dietary supply of amino acids are low, and therefore the pool is not large, the activity of these enzymes will slow down. And our last scenario is for a high protein diet. So in this case, you're well fed, but you've got very high levels of intake of dietary protein. So hence I've got a big fat red arrow here. Because you're well fed, your body's turnover of protein will be relatively constant. So you'll be synthesizing and breaking down body protein at about the same rate. And therefore, to balance out this big red arrow here, we need a corresponding increase in the rate of deamination to maintain that amino acid pool at a constant level. And therefore, under high protein diets, you deaminate a lot of amino acids, producing glucose and also fat. And in addition to that, the excess nitrogen is synthesized into urea and excreted. Now, I quite like this story. I'm sure you've all got a friend that probably goes to the gym and lifts lots of weights, and they may be mucking around with amino acid supplements in their diet. Now, to be honest, the average Australian diet will give you all the amino acids that you need to maintain a good nutritional intake to fuel your gym work and your big heavy muscle growth. So have a guess what happens to the extra amino acids that you might take as a supplement. You probably bought them for $200 a kilogram at the local chemist, but they're effectively doing this. You've got increased dietary protein intake, massively increased rates of deamination, more fat being synthesized, and a lot of urea being produced. So in effect, the bulk of those amino acids are being pissed out as urea and stored as fat. Not a good outcome. And of course, the control over this process, again, is very simple. It's through the Km of the enzymes involved. So the transaminases and deamination enzymes involved in this step here have a high Km and therefore become very active when dietary protein intakes are high. In addition to that, the enzymes of the urea cycle have a high Km, but they're also increased in expression under high dietary protein intakes, enabling your body to handle the increased rate of excretion of nitrogen. So in conclusion, in this lecture we've looked at how individual amino acids differ and in particular how the R side chains differentiate them. We've identified that there are 10 essential and 10 non-essential amino acids, making a total of 20 that your body consists of. We've looked at the lysosomal and ubiquitin proteasome pathways for protein degradation, and lastly we've discussed the concept of nitrogen balance. And to help you recap this lecture, there's a series of learning objectives that you can go through to make sure that you understand the material.